Hello friends, today's topic is excretory system. Introduction. Excretion is defined as physiological process in which nitrogenous waste material that arises due to metabolic reactions are removed from the body. Excretion simply mean removal of nitrogenous waste materials. The major nitrogenous waste material is ammonia, which arises due to deamination of nitrogen containing compounds such as nucleic acid, protein. The ammonia released from deamination is transported to liver and converted into less toxic form called urea by a cyclic process called urea cycle or ornithine cycle. Human excretory system. The excretory system of man consists of a pair of kidney, pair of ureters, an urinary bladder, and an urethra. The accessory organs are lungs, large intestine, liver, and skin. Human kidney. A pair beings of structure and metanephric in nature. Each kidney is located at the either side of vertebral column. Kidney are located at the level of T12 to L3 of vertebral column. The right kidney is located slightly lower than the left kidney. The kidneys are attached to the dorsal wall of the abdomen by peritoneum. Histology of kidney. Human kidneys are retroperitoneal organ. Kidney is surrounded and protected by three layers of tissue such as renal fascia, outermost layer made up of fibrous connective tissue, then adipose capsule, medial layer made up of adipose tissue, then third one is renal capsule, inner layer made up of fibrous connective tissue. Then function. Provides mechanical protection. Holds kidney in its position. Checks spreading of infection. Structure of kidney. The human metanephric kidney is differentiated into outer cortex and inner medulla. Medulla. Inner portion and it constitute about 80 to 85% of kidney. The funnel-shaped structure is called renal pelvis. It acts as collecting space for urine. The door of the kidney or the opening is known as hillos. Each kidney contains 7 to 15 minor calyces and 3 to 4 minor calyces, united to form one major calyx. And the major calyx opens into the renal pelvis. Each kidney contains 7 to 15 conical shaped structure called renal pyramid. Renal pyramid opens into minor calyx through papillae. Cortex. It is the outer portion of the kidney. It is more darker and granular than medulla. Due to more blood supply and coiled portion of the nephrons, some of the cortical tissue has embedded into the medullary region on between pyramids to form renal column of Bertini. Nephrons or uniferous tubules. These are structural and functional unit of kidney. Each kidney contains approximately 1 million nephrons. Each nephron is highly coiled structure and consists of glomerulus, Bowman's capsule, proximal convoluted tubule that is PCT, loop of Henle, distal convoluted tubule DCT, collecting tubule, collecting duct, glomerulus, bunch of capillaries that is 20 to 50 afferent arteries located in cortex. Coiled in Bowman's capsule, specialized pores are present called fenestrae, which increases the permeability by 200 times than other. Then second one is Bowman's capsule. Located in cortex, cuff-shaped structure that encloses glomerulus. The wall is of two layers. The outer layer, that is simple squamous epithelium. Inner layer is lined by specialized cells called as podocytes. Podocytes with finger-like projections called pedicels. The space between pedicels are called filtration slit. Bowman's capsule and glomerulus together form Malpighian corpuscles and renal bud. Third one is proximal convoluted tubule. Located in cortex, highly convoluted part, it is lined by cross-border cuboidal epithelium. In epithelium containing microvilli, it increases the surface area for the absorption. Then fourth one, loop of Henle. It is long U-shaped tube-like structure located partly in cortex and partly in medulla. Both the limbs are lined by simple squamous and simple cuboidal epithelium. However, descending limb is permeable to water and impermeable to solutes, and the ascending limb is impermeable to water and permeable to solutes. Then fifth one is distal convoluted tubule. It is located in cortex, lined by two types of cells, light cells and dark cells. Light cells, less in number and contains microvilli. Dark cells, more in number and contains pigment melanin or macula densa. Collecting tubule and collecting duct. The DCT opens into thin tube-like structure called collecting tubules. The collecting tubules open into a duct called collecting duct. The collecting duct opens into a large duct called duct of Bellini, which opens into minor calyx. The collecting tubule and duct are lined up by two types of cells called I or interskeleted cells or P by principal cells. When filtrate reaches collecting duct, it is called as urine. Ureters. A tube-like structure arises from pelvis of kidney and opens into urinary bladder ventrally. Then the wall of uterus are made up of three layers such as FCT, fibrous connecting tissue, smooth involuntary muscles, longitudinal layer or circular layer. Then transitional epithelium, that is urothelium. Persistabile is carried out by smooth involuntary muscles. Then urinary bladder. Urinary bladder, it is a single layer pear-shaped organ located in urethra. Function. It helps in temporary storage of urine. It also helps in voidation of urine and only urine in case of female. Male and female urethra differentiation. Male. It consists of three regions such as prostatic, membranous, penile. Length 16 to 20 cm. Carries both urine and serum. Female. No such distinct region is found. Length is 4 to 6 cm. Carries only urine. Types of nephron. Cortical nephron. It is present deep in the cortex. 85% of kidney covered by cortical nephron. Loop of Henle is very short. Juxtaglomerulus complex is absent. Rate of filtration is 1000. It works in normal condition. Then second one is juxtamedullary nephron. It is present at the junction of cortex and medulla. Is present in 15% of kidney. Loop of Henle is well developed. JG complex is present, rate of filtration is high, it works at the time of strain shortage of water. Juxta glomerulus complex or apparatus. JG complex is found at the junction of macula densa cells of DCT and juxtaglomerular cells of afferent arteriole and joined together by mesangial cells. The juxtaglomerular cell secretes erythropoietin, which increases synthesis of RBC, and also secretes renin, which increases BP. It is called as endocrine part of kidney. Kidney of vertebrates. The primitive ancestral kidney seen in cyclostomes called as archinephrons kidney or holonephrons. It is differentiated into three types: protonephrons kidney, mesonephrons kidney, metanephrons kidney. Protonephrons kidney, anterior part of archinephronous kidney, it is not well developed, not differentiated into cortex and medulla. Glomerulus is absent, number of nephrons is less, found in adult of cyclostomes, that is myxine. Then second one is mesonephronous kidney, middle part of acronephronous kidney, glomerulus and Bowman's capsule may be absent, cortex and medulla absent. Number of nephrons more, found in cyclostome feces amphibians. Then third one is metanephrons, posterior part of archinephrons, highly developed glomerulus and Bowman's capsule except reptiles. Cortex and medulla present, number of nephrons very high, found in reptiles and man.